Thank you for taking the time to be with us here today. My name is Akil Pulapati, and today I'll be presenting alongside Michael Amadeo and Probian Carr. Also here today are team members Farah Vazic, Asana Darwish, Eric Lai, and Abriana Walls. This is Team Blood's Thesis, Proposal Defense.
Akil, take one little step to your left, please.
You got it.
Thank you.
All right, so really the genesis of our project and the inspiration is acute myeloid leukemia. AML affects 36 out of every 1 million people, and what this malady is it happens when hematopoietic stem cells don't differentiate properly and causes leukemic blasts to overcrowd normal blood cells, which causes infection, it can cause bleeding, and can cause death if it's not treated properly. Now, current treatments for AML include chemotherapy, which totally and drastically reduces the number of red blood cells a patient has. Now, this kills healthy blood cells, yes, but it also kills off the leukemic blasts. Then, in order to reinstate healthy blood cells, because the patient just lost a bunch of red blood cells, there's a hematopoietic cell transplantation. Now, the problem with this is that graft versus host disease is a real problem, and this happens when the donor cells are rejected by who is getting the cells because of an immunological mismatch. So the ideal treatment would take the patient's own cells, and this way we could avoid graft versus host disease. Now the best method we believe to do this is induced pluripotent stem cells, because this would take a patient's own cells and reintroduce it back to their own body. So it would take a somatic cell, like a skin cell, and we introduce it to an array of transcription factors to induce it back into a pluripotent state, a state in which it can become multiple cells. It can become a blood cell at that point, it can revert to a skin cell, whatever that you want. So this method, iPSC, it's a new field that's up and coming, and we feel that it can truly revolutionize the graft versus host disease problem, because through iPSC we can really avoid that. So Team Blood, what we're doing is we're trying to test these iPSCs to see which ones are best at hematopoietic cell differentiation. So we actually have two aims. The first one is to produce iPSC through 3 different methods that we have looked up and researched during our literature review. After we've obtained these iPSCs, we're going to put them through another method to create the hematopoietic stem cells. Next slide, please. And here you go. You can see what our goal is. We kind of laid it out here. So we're going to take peripheral blood, introduce it to an episomal plasmid treatment to create the iPSC. The other 2 methods we'll use fibroblasts and introduce them to a lentiviral treatment and transposon treatment to come up with iPSC. Now, the reason we chose these 3 methods is that after conducting our literature review, we felt that these were the most feasible for us to really compare, as well as the most impactful for the field of science. There's thousands really of methods out there, but these 3 are the ones that we really feel that we can test in a significant way. After we have these iPSCs, we're gonna, like I said, introduce them all to an OP9 co-culturing method to create the hematopoietic stem cells. And after we obtain the stem cells, we're gonna test them both in vitro and in vivo to see which method was most efficient. Now Michael's gonna talk more about each method specifically. Michael.
Sure. So as Akil mentioned, we have 3 methods in our phase 1 of our project, which is getting iPSCs. The first of which is our peripheral blood method. Now, this method converts peripheral blood mononuclear cells to induce pluripotent stem cells by centrifuging blood through a concentration gradient, or without a density gradient, excuse me, and then we plate them onto a dish to allow for growth. Now, after a period of time, and after we observe growth, we will then transfect these using episomal plasmids and then plate them onto a dish containing mouse embryonic fibroblasts. After a period of time, which is approximately 5 to 10 days, we should begin to see reprogramming in the cells and a lineage of induced pluripotent stem cells, which we will check by immunofluorescence and gene expression studies. Our next method in phase 1 of our project is our fibroblast transposon method. And essentially what this is, it's introducing a vector of transcription factors to the genome of a somatic cell to induce pluripotency. And so first what we'll do is we'll purchase a transposon vector, which can be bought from any lab, and we will then encase it in a liposome. And as you may recall, liposomes are positively charged particles, and when they bind with the negatively charged cellular membrane, they deliver their contents, in this case our vector of transcription factors, into the cell. And after that happens, we should be seeing reprogramming in the cells, And to test for pluripotency, in this case, we're going to be using immunostaining to test for the nano-G gene, which is actually a gene that has been highly associated with reprogramming in somatic cells. And after we test for the nano-G gene, we're actually going to be removing a transposon because this has been shown to potentially be an issue later on in our project when we move to phase 2 of our project. Our final method of phase 1 is our lentiviral reprogramming method. And essentially this converts fibroblasts to iPSCs using lentivirus equipped with transcription factors. So after we infect the cells with a lentivirus, we then apply doxycycline, which has been a proven gene expression inducer. And after we do that, we will then treat them with glutamine, DMEM, which is Delbeco's modified eagle medium, and then fetal bovine serum. And once we do that, we should exceed these ambroid bodies over here. And essentially what that is it's a aggregate of stem cells, but not quite the stage we want yet. But eventually, after a period of 5 to 10 days, they should become the iPSC line that we want. And once we have this line, we'll check the cells, see if we got the yield we wanted, and then repeat differentiation of experimentation if this yield is not up to par with what we expected.
So all the cell types are mouse? So that you're starting with peripheral blood and fibroblasts, those are all mouse?
Correct.
And the feeder cell lines are all mouse?
Yes.
Okay.
Yes.
Did you use for the first two methods?
Correct.
Okay.
Correct. So for phase 2 of our project, we're going to be taking these 3 previously produced stem cell lines and inducing hematopoiesis in them. So the method we chose to do this is by taking our iPSCs and culturing them with a line of mouse cells called OP9. Now, we chose OP9 because it's both quick and efficient. It actually only takes about 6 to 10 days to get significant reprogramming, and it also does not require exogenous growth factors to work as well. So after we co-culture the two together, we will then be adding a growth medium, and this should induce hematopoiesis in the stem cells to give us hematopoietic stem cells. And after we collect those, we'll then be moving on to the data collection analysis of our project, which Purpling will be talking to you about.
So throughout our research, we're going to be collecting our data in 2 methods, and this will analyze it both qualitatively and quantitatively. First, since we're essentially producing our own cells, we need a way of verifying that we actually produce the cells that we think we have. And the way we're going to do this is with immunofluorescence checks. So each cell we produces antigens that are specific to that type of cell. And so we introduce antibodies to the cell, which bind to that antigen, and then we introduce a secondary antibody, which is tagged with a fluorophore, which binds to the primary antibody. And if we are able to view these cells under immunofluorescence, we can conclude that the antibodies have bound to the antigens, and the antigens have been produced, and thus we can concur that we have produced the cells that we think we have. Next, after phase 2, when we produce our hematopoietic stem cells, we will be testing their engraftment potential in mice. And so we will isolate our HSCs and inject them into mice, and after 60 days we will isolate their peripheral blood, their lymph nodes, their spleen, and their bone marrow, and we will undergo that in flow cytometry. And what the flow cytometer will do is it will sort them between cells that have the CD34 marker and the cells that do not. And the CD34 marker is a marker of hematopoietic stem cells, and it will give us a count of those cells as well. We hope to see that one of our methods of producing iPSC and then later inducing hematopoiesis is more effective than the other 2, and we're just measuring this by the number of cells, the number of viable cells that we get. And we anticipate that this will be the peripheral blood method. And that's because the literature that we have read leads us to believe that iPSCs that are produced from your desired end source and then reprogrammed again to that end source will be more effective than iPSCs that are produced from a source that is different than your end source. This is just a brief timeline breakdown of where we hope to be over the next few years. By the end of this semester, we hope to finalize our project and start applying for grants and obtaining our necessary reagents. By this fall, we will begin our lab work and start data collection for phase 1, and we'll also be preparing for junior colloquy at that time. A year from now, in spring of 2017, we'll finish phase 1 and start phase 2, and we will also present at undergraduate research day in the spring. The fall of our senior year, we will finish up our lab work and data collection, and we will start preparing for senior thesis conference. And then our final semester here, we will complete our team thesis, and we will prepare for defense at the team thesis conference. This is a budget breakdown of our project. As you can see, most of the cost comes from our reagents, about $7,270. This is under the assumption that none of our reagents are available to us on campus and that we will be buying all of them ourselves. Our mice are about $1,160, and the flow cytometer accounting for about 10 hours of use is at $400, for a total of $8,830. Now with all those costs, of course, we're going to have some limitations and obstacles, and two of those are funding and obtaining supplies. Hopefully we can address those with funding and grants and such. Another limitation to our project is that we will be using a limited number of methods. We're only testing 3 methods that we believe are the most popular, the most effective for us to use, and that are most efficient for using blood, but there are many methods of producing iPSC out in literature. If we wish to work with mice, we will need IACUC approval, and of course, since we are working in a microbial lab, we will have to be very careful about contamination. Now with that said, there are still plenty of things that can go wrong in science, because science is never black and white. So we've created a plan B in case anything goes wrong anywhere within the scope of phase 1 and phase 2 of our project. In phase 1, if we're unable to produce iPSCs, we can address this in a number of ways. And one way we can do that is we can purchase our own iPSCs. And when we purchase them, they will tell us the source and the reprogramming method that the cells have been made from. The only downside to this is that we're not able to control for the environment in which the cells are produced. We can also try to go back and troubleshoot and see where we went wrong. And if time and budget permits, we can see if we can find an alternative methodology that might work as well. If something goes wrong in phase 2 and we're unable to produce our hematopoietic stem cells, again, we can troubleshoot and try to see where we went wrong. And if time and budget permits, we can go back and also try to find an alternative methodology that might work. We would like to take this time to acknowledge our mentor, Dr. Nam-Sung Wang, our librarian, Ms. Natalina Changalova, the Gemstone staff, Dr. Cole, Dr. Skendel, Ms. Lee, and Ms. Hill, and of course our experts, Dr. Fisher and Dr. Stroka. Thank you so much for being here with us on this wet and windy morning, and especially for the valuable feedback you will provide us on our project. Any questions?
Excuse me, great presentation. We have a tradition to let our external experts start with the questioning. That lets us off the hook, really. So you can get to decide who would like to go first.
Okay. So I think it's a really neat idea. My first concern is it's very ambitious. There's a lot of steps. And have any of you done cell culture before?
I believe one of our team members has.
It's not
He's not here right now.
Okay. I don't know if you agree or not, but
It's gonna be a little challenging.
yeah. So that's just my first comment. So you guys are aware that you have a lot on your plate.
Yeah.
Right.
So can you guys talk more about so for each of the different methods you're planning to use in the first aim, so you're planning on using immunofluorescence, right, to try to see whether or not you have your iPSCs. Can you talk more about the specific markers for each of the different methods?
Right, so we talked about the nanogene that is produced in some of the cells, and I'm not sure what the other one was in the other iPSC cell marker, but they have their own.
So is it known that if it has specifically that marker, it has to be an iPSC cell, or?
Yeah.
Okay.
So they're unique.
They're
markers.
unique to that specific
To
cell,
iPSCs.
yes.
Okay.
Okay.
So if you find it, you have it.
Yeah.
Okay.
So what percentage of cells are you expecting to have?
Right, so the methods
To become indisciplined.
Right, they greatly vary. In the literature review, sometimes it's like less than 1%. So honestly, that's kind of part of our project because like we have all these different methodologies that are out there in the literature, and so we don't expect necessarily like a great amount of yield, but we want to just compare the yields that we get from each method, and it's not necessarily going to be that high.
So could you take a guess of what you might expect based
Well,
on what
I
other
know-
people have found?
At least preliminarily, we expect peripheral blood to be, I think we talked somewhere in the neighborhood of 10%, which I think could be ambitious but possible. The other methods I mentioned actually for the lentiviral, they've been shown to not be as effective. So I think for those, I mean, like Akil mentioned, less than 1%. Those are just raw numbers, but I think definitely we expect peripheral blood to be the most effective.
Jump into the comment here. All the team members not only should but are expected to participate. So if you feel uncomfortable fielding questions sitting in your chair, stand up. If you feel comfortable stand up and fielding questions, sit down. Just be part of the conversation.
how do you guys plan on breaking up the work?
Well, I think we plan on having sub-teams, so I know at least a couple of members are interested in working in the lab. I know Farah mentioned she was.
Yeah, we want to keep the people in the lab like
To a minimum,
down so
yeah.
that there's less variability and like
Yeah.
less chance of error between people so that it's consistent. So we were thinking like 3 to 4 people working on the first 3 methods, like maximum out of the 12 that we have.
The people that are working on the methods are also not going to be comparing with each other so that we can eliminate bias and see.
Okay. So, like, you'll have one team working on the first method and one
Yeah.
team working on the second method. Okay. So what happens if only one of your methods ends up working and you get iPSC cells from one of the methods, but then the other 2, you can't?
Right. Well, I guess we'd have to report that first of all. We'd be like, this is the method that we got iPSCs from. We didn't get iPSCs from the other 2. Then we'd have to go to Plan B, which would be to purchase the iPSCs. So it would be, another expensive endeavor. But that's really the option, like the course of action that we'd have to take at that point.
Mm-hmm.
Can you scroll back to the very first flow diagram of how you created the cells and how you, that animation's great, but go back further.
This one?
That one, yeah.
Right.
Okay, just, now continue. I just want to
Just
stare at
wanna
it a
look
little
at
bit. Okay.
And it'd be confusing.
Yeah, so I mean, I guess I'm going back to the feasibility issue, and there are a lot of methods that you guys will have to learn throughout this. Do you have the resources to actually learn the methods that you'll be using?
Right. Since we're actually pulling from methods that are already in literature, the methods are all actually already available to us.
Okay.
You still have to learn them.
Yeah.
Yeah,
Yeah,
right.
we do have
Just
to.
that's it, yeah.
Right.
We do.
Right.
And they never work. It's not like a cookbook, the cake
No.
flops.
Right.
First time it flops.
And also, some of the methods may be available in one visual journal that is called Journal of Visualization Experiment, JOVE, and it will be easier for you to just look at the videos and learn some of the methods
Right.
how to do it in the lab. So,
Thank you.
hopefully this will
Yeah,
help.
that'd be
Yeah, it's a great resource.
Yeah. So are these, because for the iPSCs you can grow them either as spheroids or as monolayer cultures, right? Sure. So do you guys know which method?
I don't. Does anyone else have any input on that? I don't know if we actually looked into that.
Can
That's
you
a good
repeat the question? I'm sorry.
question.
Yeah, so you can either grow them as spheroid cultures or as monolayer cultures. And I think the spheroids tend to produce, or, oh, you don't know? I think they're easier to grow as spheroids than monolayer cultures.
Yeah, that's something that.
But then the way you detach them from the surface, like from the feeder cells, also depends on whether they're monolayer or spheroid cultures. So there's different reagents you can use to detach them.
Right.
So just kind of keep that kind of stuff in mind.
Thank you.
Thank you.
I've never worked with iPSC cells, but I'm trying to learn about them and recently wrote a grant. So
Yeah.
Alright,
Well-
Fish, you want to continue?
Yeah, I mean, I'll just, from what Dr. Stroka said, first of all, I think you guys put together a very nice document. It was easy to read. Things were laid out and organized, and I think that's a great start. And I think I did a nice job identifying like a target problem and a good, reasonable solution to that problem. So I think that's also an excellent start. The first thing I would say is there's a great resource that you should explore, and it's a fellow named Kurt Siven. Dr. Stroker knows him as well. Our lab works with him. He was at Hopkins for a long, long time and then moved to Maryland about 8 or 10 years ago. He's the director of the Stem Cell Center, and Kurt's lab was the one that identified CD34 as the marker for HSCs. And his appointments are in both cancer and pediatrics, and he came up with the clinical strategy for using HSCs to treat pediatric leukemia. So I would take a field trip contact him, and if you need his email, I can send you his email.
Thank you.
And contact him and take a field trip up and talk to him. He knows way more. I won't say anything about Dr. Strogo. He knows way more about this than I do. So I would use that resource. So his last name is Sivin, Civin. He's Associate Dean for Research at the medical school. Then also picking up
Shut the door, please.
what Dr. Stroka said, yeah, I'm pretty concerned that it's ambitious. Now, these guys knows I've worked with 3 different gemstone teams, so I understand everybody's excited and eager and wants to do a lot, but I'm pretty concerned that you can really master 3 different techniques just to do the first half of the project. So I would maybe think about how you could, and I don't want, I mean, you've worked with your mentor, and I don't want to change everything for you guys, but I would think about how you could narrow stuff down significantly, because I think it's going to be challenging, just as a broad comment. Then go to more science. You're comparing a method 1 that starts with peripheral blood cells and 2 that starts with fibroblasts.
Right.
Is that a straight up fair comparison? Because you're starting with different cell type. Cells have different propensity for genetic manipulation.
Absolutely.
So it may be that just fibroblasts are more amiable to the 4 transcription factor approach for making iPS cells than the peripheral blood cells, or it might be the other way around. So like as like an experimental design thing, since you're starting, now you said you're using all mouse cells, but since you're starting with distinct cell populations, you're not just comparing the methods, you're also comparing your starting cell populations, right? So that might be a challenging comparison to make. So I would think about that. One suggestion to pare it down would be maybe pick one method. One method's gonna be hard to figure out. I would pick one method and maybe see, could you make it more efficient by tweaking the culture environment or all these things. There's a fellow who was at Georgia Tech and he's since moved to California, and he did a very simple thing where culturing these emboid bodies, he said, well, emboid bodies, they're in suspension culture. His whole lab basically is set up on the idea, I'm gonna culture these on a shaker table. That was his advance. Do you know this, Todd?
Yeah.
He's like, we'll do it on a shaker table so everything's kind of stirring. And that was like a significant development to culturing. He was doing embryonic stem cells. So it can be a very simple thing, but it may be preferable to pick one method and try to develop that method than trying to do all 3, because it's going to be extremely challenging. So I would think about that. Then you said you wanted to define what's the most efficient.
Right.
So how do you define most efficient?
So we chose these methods because they're fast and cheap in that sense, but also efficiency in the sense that they produce just the most amount of viable hematopoietic stem cells.
But you're starting, like with peripheral blood, you're starting with a very heterogeneous cell population. So efficient means if I have one cell, if I have 10 cells, and
Right, like the
five
% yield.
becomes
Yeah.
iPS cells. But peripheral blood, it's not that you have 10 cells that are all identical. You have 10 of a very heterogeneous population. So to define efficiency might be challenging. Now fibroblasts a little bit because it's more of a homogeneous cell population.
Right.
So that may be something to think about. Then in the methods themselves, like as Dr. Soshi said, I think you have the steps are really well defined. I think, I mean, you're gonna have to figure out a lot on cell culture, but I think the steps are in good shape. One thing that's kind of lacking though from the proposal is an experimental design. So, what's your control group?
Right.
What would be a good? Can you tell? I mean, I don't think it's in here now, but could somebody tell me like what would be a good control group? In the first part where you're trying to develop iPS cells.
Good question. I honestly, our game plan essentially was to just go with for these 3 methods, go with what they did in the literature. I don't remember ever reading anything about them using control groups, but like, yeah, that's something to look into.
Especially since
We
we're
haven't
producing them all in
thought
the same environment. I agree
Yeah.
that we should have a control for a basis of comparison.
Well, you can buy IPS cells, so you can maybe compare your
That's
cells
a good point.
to those
Purchase.
cells. You can get IPS cells from other labs. I think we have some in our lab. I have to check, but I think we have some frozen someplace in our lab. So those might be good groups to compare your work to. And then in terms of the assays, so you're going to use the surface marker approach to say that they're IPS cells at the end of the first step. What's the real experiment to demonstrate that they're IPS cells?
Well, it's interesting because I didn't think we had really advanced our, knowledge on the topic enough to know that there would be any way to identify them other than just using immunofluorescence, for example, and looking for these these genes in it, and that kind of method. But it's interesting that you bring that up because I don't think we'd really discussed that in
I
our
really talked about that. Yeah.
Well, so they're supposed to be like embryonic stem cells. And what's the phenotype of an embryonic stem cell? Like what's its main characteristic?
That's pluripotent.
So it's pluripotent. So what's the real way to show that?
I mean, to like truly show that, like if it could differentiate into like different things, then that would be.
So how do you show that?
Yeah, that's a good point though, because if we're just going to iPSCs, then we need to then prove that they're right. You know what I mean?
Right.
Right.
That's a good
How do you
point.
So
demonstrate-
how do you demonstrate pluripotency? The real way to do it. I'm not saying you should do it. You probably really don't want to do it. But what's the real way to do it?
Differentiating them into all the different.
So
cell
tell me how you
types.
do that experiment.
Adding like exogenous like factors like cytokines or other things like that. Or
No. Taking those cells and
Yeah,
putting
check.
them into a blastocyst, marking those cells and putting them in a blastocyst, let that blastocyst grow up to a mouse, and then checking to see if all the cells, all the tissues in that mouse
Aren't
have your marked cells. That's the real way to demonstrate that an embryonic stem cell is an embryonic stem cell or an IPS cell is an IPS cell. Now, what you're doing is completely reasonable, but I think you should be aware that ultimately you're just measuring markers, which
Right.
are markers, but you probably want to. You could even just do a simple in vitro assay to try to differentiate them down a few different lineages and show that they actually do differentiate down in different lineages. Like our lab does a lot with mesenchymal stem cells, so the simple assay there is show that they become bone cells, cartilage cells, and fat cells. So just those three lineages. So maybe you might want to do something similar in vitro, just to show those cells can become a couple different fully differentiated cell types.
Thank you.
Thank you.
That's a very good point. Thank you.
Then, yeah, I guess lastly, yeah, I think every gemstone team has like a different model of how to work. You guys have 11 members, so how many people are going to work in the lab?
We said a max of about 3 or 4 for each method,
Oh, for each month. Okay.
right. And then for the next part, the OP9 co-culturing, we said about 2 people, because we want to limit it in terms of like contamination and consistency.
Is everyone going to be around all?
Most of us
all
live
years?
in the
Yeah.
state. Yeah.
No. I mean, like, is anybody going to go abroad or?
We
Summer
discussed it, but we would
internships or-
definitely pick the people who weren't gonna go, and
Okay.
like we would have tasks for them to do while they were abroad. To
Yeah.
Like, keep up on literature and.
And are people going to try to work over the summer?
yeah, we definitely plan on it.
Good plan.
Is that really gonna happen? Because, I mean,
Everyone's
everyone's like, yeah, sign me up.
Yeah.
And then it cuts to be May, like, no, I got a job, or I got an internship, or I wanna do this, or I wanna do that.
I think we will. I mean, I know personally, I'll probably be on campus this summer, so and in future summers as well. So I believe we'll have people to do it.
Okay, yeah. Well, we used to always try to see if we could finagle a way to pay somebody, because people often need money, but you're not really allowed, right? Or can't, or is it, rules change?
It can be worked out if
Okay,
they,
worked
if they
out.
have the funding and if we can talk the mentor into supporting that.
Oh, I didn't mean to do that. I was trying to pony up somebody else's money.
No.
Yeah,
It depends on how the team wants to spend their resources.
right, yeah.
Yeah, I think it's important to, because during the semester you guys are going to have classes, you're going to have other commitments, and it's going to be challenging, I think. I mean, you have to do it because you have a limited amount of time, but to do all the cell culture and to get all the methods done, I mean, it's going to be hard to work in between all of your classes and everything
Right.
to so like strategizing that. So having big blocks of time during the summer when you have full days to work, I think that will be
Yeah.
helpful for you guys.
And yeah, I mean, just to follow up on that, you're gonna need a very, very efficient way. Someone's gonna go in and start something on Monday morning, and then somebody else is gonna go in Monday afternoon to try to pick up from that. And you need a very detailed way to make sure the person who comes in the afternoon can pick up from what was done in the morning and not mess it up, because it's gonna happen. So, yeah, or you try to find a schedule where all three people of that subgroup can go together, but that's almost like nearly impossible to do. Yeah.
All right, let's see, Natalie, you want to ask some questions?
Oh yeah, this is something regarding the writing and your final thesis. I know it's not on your mind right now, but just to avoid copyright violations without the images that we'll be using, it takes some time to take some permission from the publishers, and here is a copy from the APA citation style book, just as an example how you have to cite your images. And in many cases, you have to ask for permission, and it may take some time to get it. So take this in terms of for your final thesis.
Thank you.
Thank you.
And to follow up on that, if any of the images are original that you've created, give credit where credit's due. Therefore, there's no question that it has not been cited. So whoever made it, give them credit right there on the image.
And also, I noticed from other Johnston teams as well, they consistently use images from the free internet, although they are from reputable websites, like from example, the National Institute of Health and the CDC website and so forth. Try to find images from the scholarly literature. It will give more credit to your final product.
Thank you.
Thank you.
Amy, you have some questions you want to inject?
Yeah, can you go back to that method one, the one you just
Maybe.
flashing at the end?
Did you try like unplug it and unplug it?
Yeah.
It's the physical.
Right, yeah. It's the one, right there. What happens to the plasma you put in? What happens to the virus you put in?
I believe the virus actually acts, it basically infects the cell, and then the cell becomes an iPSC, and then at that point, I think we determined that the virus wasn't in the iPSC anymore. After it infects the cell, because it had already infected the cell with the transcription factors.
You just
Look
get
at
rid
what-
of itself. It's not gonna stay around.
Okay, yeah, I mean, I guess that's something to definitely look into. I mean,
No.
Okay.
I didn't think that, it didn't occur to me that, the virus could still be in the cell after it infects the cell, which seems pretty common sense actually, but, yeah, we'll definitely look into that.
No, iPSC, I think they were mentioning that how many passages, how many times do you want to multiply them? Out of, let's say, 100 cells, let's say if it's 1%, you get one iPSC. After you get one, you want to multiply that, right?
Yeah.
Absolutely.
Okay, how long are you going to multiply that?
I-
By the way, I think that's also another way of checking whether they are, that they're any good, because if it's iPSC, you should be able to multiply forever.
That's true. I think it's basically gonna come down to whatever is an observable amount, which we can look at under a microscope and, as Dr. Fisher mentioned, maybe do some testing to, put in a blastocyst, something like that. I think number-wise, I think we just want enough that we can.
Right. Well, I think
You know what I
the
mean?
key is for us to have like enough so that for the second stage of our project, when we have OP9 co-culturing and then we test it both in vitro and in vivo, we have enough from all three methods to do that. But then of course we have some left over because they are iPSC. We can always just make more.
Right, but then on the other hand, so for example, as a control, do you think it matters if, let's say, a source of iPSC that's been passaged around 10 times versus another one that's been passaged around
Right.
20 times?
I think, yeah,
Because
we
you gotta
definitely
feed
have
these
to
guys
normalize
when
that
there's a
somehow.
passage, because you
Yeah.
gotta feed these guys, you gotta change the nutrient, you gotta change the media periodically.
Right. We have not thought about that.
We have to think about whether we
Yeah.
want to normalize it to the amount of times you reproduce or the number of cells that you.
Can I make a quick comment too?
Mhm.
I don't know about iPSC cells, but I know other types of stem cells, if you get up to a certain number of doublings or passages, they lose their, they start to differentiate. So
Right.
I would just, I don't know,
Look into that and
yeah,
Okay.
be careful with that.
Is that a fact that could be easily determined from the literature? You can replicate them X number of times, or there's an average of number out there that, or you have to just
Yeah.
flat out guess, trial and error?
I'm sure
I'm sure it's out
it's in the
there,
literature, we just haven't looked into that aspect.
In theory, an embryonic stem cell and therefore an iPS cell can double without limit. They're the only normal cells, non-cancer cells that can do that, but in practice, that's not really the case.
Okay.
I mean, that's supposed, there are three main phenotypes: pluripotency, limitless proliferation, and you know, they have to have, when they differentiate, when they divide, they create a daughter cell of a different phenotype than the parent cell, so that you can repopulate the parent stem cell population. So,
More questions, sir?
Basically, that has some implications, because let's say you start with 100 cells, you end up with one iPSC, and if you can only double so many times, let's say you can double to 50, you actually lose. You start with more cells than what you end up, right? And you know, with the next step, let's say you lose another, I don't know, certain fraction, you actually end up with even less number of cells than when you started. So the whole point here is to actually get more, right? So you have to be able to multiply your iPSC.
Okay.
How about the ratio of this iPSC and OP9 cells? Does it matter? How you co-culture these
Eric,
things?
There's a specific, well, when we get the OP9 cells, they have, they give us a culture that we grow the OP9 cells, and I'm pretty sure there's a specific number of iPSCs that we should culture it with. I'm not sure exactly that amount, but I'm pretty sure there is a ratio. But
Do
I'm
you
not.
need to separate them out later afterwards? Do you see a need for that?
I'm actually not sure. They, once you co-culture them, they should, can you go down to the OP9 cell?
And you said they both grow and you got a mixture, so what are you gonna do with the mixture?
Right. Hold on. Once you treat them with the growth medium, you should be able to harvest just the iPSCs, the HSC cells. So like, there probably will be some that aren't like the OP9 cells left over, but there also will be the differentiated iPSC cells. So we would have to extract those from the mixture.
How do you do the separation extraction?
Not sure.
Okay. How do you know your iPSC isn't cancers? Because they are really those two types. They are very close. I don't even know what's the difference between them.
Right.
I guess that would go back to the immunofluorescence. I'm assuming we could probably tell the difference between the
Not
two.
just that, we can, the in vivo testing.
Yeah.
That's true.
That can tell us if it's cancerous. Also, I think, I'm not sure if all three of our methods, I know one of our methods does not use the oncogenic transcription factor, which is, I think it's MYCC. It's one of them, yeah. But, so in theory, it should not be cancerous, because that's really the one that, like, the literature says is, the cancer-inducing one. But really, in vivo testing is how we would be able to tell.
I'd like to pop a couple questions in from a more novice perspective. Okay, you have starting with three original sources of cells, and they're going to be all tricky to get to the point where you can actually determine whether you have success or not. Are there any early stages in the process where you can determine that you're really running into a lot of trouble, a lot of roadblocks, a lot of difficulty transform those cells the way that you want them to transform, so you can set a hurdle that your techniques have to cross, so you can decide whether a starting cell is in or out, that technique is in or out. So you don't have to worry about laboring over all three processes if one of them's going to be a failure.
Right, so there's not really any like benchmarks, but another major reason why we pick these three methods are that they're quick. So like each method shouldn't take
What's
more
quick
than
mean?
a few weeks.
Few weeks, okay.
Right. So over the summer, if after multiple few weeks, so I guess like a month and a half.
The whole summer.
If we cannot obtain IPSCs, then we know we have to move on and just purchase IPSCs at that point.
Yeah, there's also limitations on just the fact that we're going to be getting funding specifically for this project with a number that we've pretty much predetermined. So if we simply just run out of our reagents, I mean, we might not be able to purchase more, and purchasing could also just throw our timeline off. So if we can't do it after the reagents that we have purchased, it might be unfeasible at that point.
Okay. And as far as being in the most bang for your bucks that you hope to get, if you don't get to the in vivo portion, can this still be a successful project?
Yeah, I believe so, because if we purchase the IPSCs, then the first stage of our methodology isn't really relevant anymore, but we'll have to move on to our second stage of the methodology, and we can still do in vitro testing. And I feel that through this in vitro testing and OP9 co-culturing, we can still call it a success. We can still verify that through OP9 methods we can obtain hematopoietic stem cells. So there's no failure in science.
Good tagline. Just from my experience of watching, I've never done an in vivo study in my life, but watching people do them, failure rate is astronomical. So think about your resources, think about your time, and think about how you might have a very substantial product. I mean, you may have the new shaker table. You might have a very substantial product without ever going to the in vivo study.
Right.
Okay, so bear that in mind. The success can be achieved at an earlier stage than the outline there. Just a couple comments about your presentation. I thought that the graphics were excellent, very clear. What I said in the beginning about knowing your audience, that's important. You had a lot of acronyms, a lot of very specific terminology that I think for the folks who were up to speed in that area, the experts, it was fine. I was lost a couple times, to be expected. Okay, so that's the thing about knowing who you're speaking to. If you went to more of a lay audience, like maybe a funding group who wanted to hear about this thing, you would want to take it down a notch, right, and give some more definitions. And I heard the term like grants and such. Okay, and you need about $8,000 or $9,000 to pull this off, and hopefully you're going to beg, borrow, or steal reagents from all over campus, which happens all the time. What's your plan for raising the money you need?
Well, I'm kind of in charge of funding for the team, and we were intending on doing Launch UMD as one source, but we talked to Dr. Wang, and he also believes that it's important for us to apply for scientific grants, and because it's nice to get money for free from people, but it's also making a pitch for your product is good too. So I think we definitely intend on combining Launch with some other grants, and I think I've looked at a few already through NIH.
We've compiled like a list, and we have like a list of grants that we plan to apply to.
I think there are a number
Some of
within
them, the
Maryland
deadline's
as
already
well.
passed, but-
Did you discover any that are A, locally focused, and B, student focused? So they exist.
Yes,
there
that's
was
actually-
there was like Maryland Stem Cell like Research Foundation or something. It was like extremely specific and like to like Maryland. So
That's for us. Yeah.
Okay. I mean, if you're competing on the broad stage of NIH or
We're competing with a lot.
NCR or whatever, your chances are big into those. But those locally specific or student specific programs are great. And I agree. I think grantsmanship is part of the learning how to do research process. And I encourage you to go that route. And I also encourage you to do UMD, to launch UMD, because we're really successful at this. Okay,
With
think
the
it's really,
external grant starting,
Go
Joe,
ahead.
just want to remind you that you may see the deadline is March 15th. You're like, oh, we have plenty of time. No, you don't. You're already late. So you need to make sure you're planning ahead, because you have to route anything external, including the Sigma Z grant. That's the $1,000 individual student grant. That has to go through your mentor, through his department, routed through Aura. So you need to be planning much further ahead than you might be used to with assignments for classes. And it has to all be routed appropriately, because if you don't do that, you get the money at the end, you actually don't get to use it because it hasn't been routed through the proper channels.
so the rule of thumb is you need to have your proposal ready for submission for approval process a month before it's due. So when you look at those deadlines, March 15th, you already missed that one.
And same with IACOOK too, that has to be routed through your PI, your mentor, and that needs to be done in advance of the deadline. So those are typically due, I think, on the first of the month. They prefer to get them earlier. You need to be done the month prior.
Okay. All right. You want to go ahead with some other questions?
How do you plan to hold each other accountable, especially with sub-teams and the specificity required for the lab work that you intend to do?
Right. So accountability is something that we have, you could say, struggled with, and like, it's something that we're still working on. You know, we're still working on a bunch of ideas. One idea that's been tossed around that I'm really a fan of is just keep track of how many hours that you put into the project, and you have to be honest about it, and have like some deliverables, and say like, you know, this is what I actually got done. So I feel that in the lab setting, that would be pretty productive. You said I was in the lab for 2 hours. These are the steps that I took. This is what I did. And that would be a good way to keep each other accountable, at least in the lab sense. When it comes to writing and grants and things like that, we're still working on that. We're working in progress.
To segue that question with what Dr. Fish mentioned earlier, in the lab, I mean, 2 hours, 3 hours, 10 hours, 57 hours, it doesn't matter. It's what was accomplished, what was done successfully, what failures were learned, what did you learn from those, and where did you leave the state of the science for the next team or next person to pick up on. That's documenting hours is fine if you want to, you know, check people off or did they do their job, but it's making sure that you are very accountable and have good accounting for the progress that you're making.
2 hours doing all the wrong steps is not very helpful
Right.
to you.
Right.
So making sure, particularly in the lab, and Team Medics, who graduated in 2014, ran into some challenges here, and they lost, I think, almost all of their cells from the beginning. They ended up, I think, borrowing from Team Electrode, that your team gave them cells so they could continue their research, because Electrode had successfully completed some of the early steps. So being attentive to, you know, if you say you're going to be in the lab at this time, you have to be there, because if you're not there, then the next step can't happen. And that's something that you all really need to spend some time thinking about with the calendaring and commitment to when you can do what you say you're going to do. It can't be, oh, oops, I didn't come when I said I would, so I'm going to come tomorrow. You can't do that when you're in this step of the experiments.
To have on a macro example, we had a team a couple years ago who was working with live fish, and whoever was supposed to feed the fish over the weekend decided to go to the beach or something instead. Middle of July, you know, fish didn't get fed. Monday morning, the fish were floating. Experiment over.
Right.
And ICUP protocol.
And our cook violated and trouble and all that kind of stuff. So it's not trivial. I mean, it sounds like you're gung ho, which is great, but you really got to get into those management details. We need to wrap. Any final questions from anybody here?
Just kind of going along with that, how do you guys, are you guys going to, like, how often are you going to meet, and how are you going to plan out your experiments? Because Dr. Fisher mentioned that, you know, if one of you comes in the morning, and the other one's going to have to continue in the afternoon. How do you guys plan on organizing those kind of steps and keeping track?
Well, right now we meet twice a week because it is a semester, but like you said, like it's really good idea to get a bunch of the work done over the summer. So over the summer, we haven't really talked about how we're gonna like set up who comes in when, because we haven't really started the whole process
Do you have
yet.
a place as to where you will be coming into?
And we also need a place.
Yes.
So there's a bunch of work that we need to do before summer comes.
Okay.
But yes, that's a good point. We haven't really thought about that yet.
Yeah, the biggest challenge is just to be able to grow and keep the cells alive, and without getting contaminated. That's, I think, probably the biggest challenge at this point.
In terms of organizing your task and who is accountable for every single task, there's some online software like Todoist, Basecamp. If you think it will be more efficient for you to track down the steps of your process, probably it will be easier for every one of you to keep track of what's going on.
Yeah, there's all kinds of different project management, lab management software that you may want to adopt.
I would just one tiny thing is, I mean, I think it's also important that you have some empathy for your fellow team members and understand not everybody's going to be able to devote their life to Gemstone on any given semester. One semester you might be able to do more, and the other semester you might be able to do less, and just understand you guys are all doing this, but everybody's also, I'm sure, doing other things. So yeah, just have a little bit of empathy in your expectations.
And your contributions may not be equal, but they should be fair, and fair can be divided up over, someone may be there for the summer, and then they are taking MCAT in the spring, and so they're less engaged that semester. So knowing that this is an overtime process and needing to plan in that way as well.
Just kind of a wrap-up comment. I think your project is very well laid out. Your proposal is very good. I think, from my view, I think this project has many different possible successful outcomes. I think what you laid out is an ultimately successful outcome. I think there's several places along the way you could have success. I'm not saying aim low, but I'm saying keep in mind that I think there's several possibilities for several different significant outcomes at different stages of this project. So, as you see time go by, as you see effort go by, you see excitement go by, you may need to readjust where you're going to end up. But I think there's multiple positive outcomes here, so keep all those options in mind.
I have a question for Dr. Stroker and Dr. Fisher. I know you said there's definitely a problem with feasibility in terms of our project and just having a lot to do and a lot of culturing to do. Do you guys have any suggestions on how we could alter the project possibly? I know you said potentially just making one method more efficient, but do you have any other ideas on how we could alter it to have a novel idea?
Yeah, I wouldn't do 3 different methods. I think it's too much. You're not gonna be able to figure anything out. And yeah, I would pick one method and see if there's something that you can do, or even to make it more straightforward, because just deriving IPS cells is no mean feat, or just start with IPS cells and think if there's an efficient method that you can derive HSCs from, a more efficient method. I mean, that would really pare it down in terms of things that you have to look at.
Yeah.
When we did cell culture stuff with the gemstone teams, we'd have 2 independent variables, and that's what we did for 2 years, right? So.
Yeah. Yeah. Keep it as simple as possible. And, so I agree. There's either try to make the IPSC culture better in some way, or start with the IPSC spot and try to make the HSC production better. I think that's probably the most feasible.
Just remember, any small advance of the science is tremendous, and you don't need to come out with a cure for all ills of the world with one project. Anything else to wrap up?
I guess one more kind of broad question. You guys had mentioned, so you picked these 3 methods because they have been the most successful, they're the fastest, most efficient, you've called it. And you expect the blood method to be the most successful, given that it's been the most successful in literature?
That, and also it comes from the source that we're trying to get to, so we're essentially taking it and reprogramming it into itself.
So what is your contribution to the field if it already seems like this method is the best in the literature? So what are you guys trying to add to that?
Right. So what happens is, it's like I said, all these methods are out there in the literature right now, and they claim that, hey, we have this success rate, we have this success rate, but we're trying to, what we're trying to do is put them to the same controlled environment and then see which one is actually the most efficient. Mm-hmm. So instead of just a bunch of different papers saying this is our efficiency, this is how we did it, we can at least compare them.
We're also looking at it specific to leukemia, and so we hope that maybe it could be extrapolated to other blood diseases. So maybe if you wanted to use iPSC for a liver disease, then you would assume that maybe a liver cell would be more efficient in that method.
Okay. Well, thank you, Team Blood. Well done.
Thank you.
Thank you very much.
I didn't make a lot of comments, but I made some comments today, so